Hi everyone, I will be going through what you need to include in your first lab report, which is on that lab that we worked through, light and photosynthesis. So this was in topic 2, step 6. So what you need to include in your lab report are basically 7 different things. So you need to include a detailed title, an abstract, which is about a paragraph long, an introduction, which can be anywhere from about a page to a page and a half, a method section, which for our purposes will be very short and sweet, so maybe a paragraph, and then a results section, which includes graphs, so it can be a little bit bigger just because of the figures included, a discussion section, which is approximately a page, and then a conclusion, which is usually just a paragraph. And then included with the lab report, you will have a reference section, which includes all of the references used. So we're going to go through each part of the lab report in detail so that you know exactly what to include. Okay. So first off, actually, let me go into our folder here on Moodle. And we're going to go down into our light and photosynthesis section. Okay, so our T2S5 light and photosynthesis was where we did the actual lab ourselves. Okay, so just to refresh our memories, we took a look at this lab, started on page 63. Now, remember, we didn't actually perform the lab, but we did get data from a previous class. So we're going to kind of use this information that we refresh our memory from, and the information that we collected along the way from our discussion posts to write this lab. We also, as for reference, we do have a writing a lab report section for you. This was put together by Dr. Blackwell, our department chair. He has the lab report kind of sectioned off into different sections as well. So you can also, this is a great tool to use to see, you know, what exactly goes into a lab report. So this video is more specific into what to include in this light and photosynthesis lab report. Okay, and then of course our submission section. So this is where you're going to submit your final lab report. So this will be due on April 29th. Okay, so let's get back into our PowerPoint and start with the title. So when we start with the title needs to be descriptive. So we're not just going to go with light and photosynthesis. Okay, so we need to actually write a title that is descriptive. And typically with our lab report titles, they should be about a sentence long. Now, if I look at the actual title, or if I look at the lab itself, so let me go back into the lab, I can see that it says here, the class will investigate the relationship between wavelengths, colors of light, and photosynthesis. You will write a formal lab report that incorporates all the class data for each wavelength of light. So I would go somewhere along the lines of maybe summarizing this first sentence. So maybe investigating the relationship between the wavelengths of light and photosynthesis, or the relationship between wavelengths of light and photosynthesis, something like that. So basically you're going to try to incorporate the dependent and independent variables in your title. So when we were conducting our experiment, which we didn't actually conduct, but we were looking at the different wavelengths of light. So that was kind of decided ahead of time. So that would be considered an independent variable, okay? And we were trying to see what the effect of photosynthesis they would have. So that was the dependent variable, okay? We were also looking if, the amount of carbon dioxide, so we're, looking at distilled water versus bicarbonate had an effect as well. So, you need to include the independent variables in your title. So that's why we don't want to just go with light and photosynthesis. We want to make it a little bit more descriptive in there. What types of light? Okay, the wavelengths of light, colors. Next, we're going to do an abstract. So an abstract is a summary of your experiment. Now, I usually leave the abstract to the end. Once I've completed the introduction and methods and everything, then I kind of write my abstract at the end, even though it goes at the beginning of the lab report, because it summarizes everything. So when you are ready to write your lab report, it's going to be about a paragraph long, and it just summarizes everything that is included in the lab report. So basically what you did, how you did it, and an overview of the results and your conclusions. So, what do we do? We checked the wavelengths of light, how they affected the rate of photosynthesis in those spinach leaf discs. And, we also looked at if there is a difference between distilled water and sodium bicarbonate on the rate of photosynthesis. How did we do that? We used these spinach discs and, expose them to different wavelengths of light. Overview of results, give the general, which wavelength showed the greatest amount of photosynthesis, and then your conclusions. So that should take about a paragraph. You don't want to, go into too much detail, but just make sure you cover everything. A lot of times I see students that just give like the beginning and then they forget the end, okay? Or they give the end and they forget the beginning. So you got to include all of this. Excuse me. Next, we're going to get into the introduction. So the introduction establishes the research problem. So what question are you trying to ask? Now, luckily, we gave you the questions, okay? So if we go right back into our lab here are the two questions we're trying to ask. Which wavelengths, colors of light, will produce the highest rate of photosynthesis in spinach leaf discs? We are also trying to ask, will photosynthesis in spinach discs occur faster in distilled water or a sodium bicarbonate solution? So that's the purpose of the lab, is we're trying to answer these two questions, okay? So that is the research problem that we're trying to establish, okay? So basically, these are the two questions we're asking. Next, we need to establish some scientific concepts of the lab. So what background information does the reader need to know before, we give them the results? So for this particular lab, since we're talking about photosynthesis, we need to actually talk about photosynthesis. So we need to give the reader the overall reaction for photosynthesis. So that means writing out the chemical equation for photosynthesis. So we learned about that already, remember? So we have the glucose plus the, excuse me, I was thinking I saw the respiration for a second. So, we have the carbon dioxide in the water, and then those undergo a reaction with light energy from the sun to produce glucose and oxygen. So we need to write that overall reaction out. We need to identify where the substrates come from. So what's a substrate again? Okay, that's on the left side of the chemical equation. So where does the carbon dioxide come from? Where does the water come from? How does the plant get the carbon dioxide? How does the plant get the water? Where does it enter the plant? And then describe what happens to each product produced during photosynthesis. So, how is the glucose produced? What part of the plant produces the glucose, and so forth. And then describe the roles of chlorophyll, water, enzymes, carbon dioxide, and different wavelengths of light in photosynthesis. So you might need to do some background research. Okay, so look up what chlorophyll, type of chlorophyll, and what wavelengths of light do chlorophyll use. Okay, so you'll notice just from doing the background research that there's certain wavelengths of light that chlorophyll can actually use, okay, and some that, or they absorb, and some they will reflect. So that way it'll give us a good foundation. So while you're doing this background research, make sure that you're writing down the resources that you use, and when you're writing the introduction, you're using in-text citations at the end of the sentences that include those sources. So by the end of this introduction, or this section of the introduction, you're giving the reader a good foundation of what photosynthesis is and the role of, how light energy plays a role in photosynthesis. And then towards the end, or basically at the end of your introduction, you're going to state the hypothesis and provide logical reasoning. So just to give you a reminder of our questions, so we have our two questions: which wavelengths, colors of light, will produce the highest rate of photosynthesis in spinach leaf discs? So you'll need to come up with a hypothesis. Well, which wavelengths, colors of light do you think will produce the highest rate of photosynthesis in spinach leaf discs? Okay, so you'll state which one you think it is and why. So you might know the answer based on your background research that you conducted. Okay, so you'll need to make sense of that. You will need to provide an explanation of why you chose those particular wavelengths of light. And then the second question: will photosynthesis in spinach discs occur faster in distilled water or a sodium bicarbonate solution? So you'll need to choose which one and explain why. Okay. So again, this introduction should take about maybe a page with all of that information in there. Moving on to the methods section. So with the methods section, since we did not actually do the experiment, we're just going to briefly summarize it. So in our lab manual, we're just going to give a short section that cites the workbook and notes any changes from the workbook methods, or that fully describes what you did when no workbook methods were provided. So basically for us, we're going to look at the procedure on page 64 to 65 and briefly summarize it. So let's go ahead and take a look at that. So if we look at our methods, we just basically need to kind of summarize the experiment. So we know that we're using spinach leaves. We know that we're kind of putting. We had two different groups. We had the group that tested with distilled water. We had the group that tested with sodium bicarbonate. We know that we had these spinach leaves that were exposed to different wavelengths of light for a period of time. Okay, so you're just going to basically summarize how it was set up, okay, in these petri dishes. You don't want to do like a procedure list like this. You just give a brief, couple sentence summary of how it was set up. All right. Once that is done, since we didn't actually complete the lab ourselves, just state where we got the data from. Okay, so where did we get the data from? Well, let's go back into our Moodle page. Where did we get it from? We got it from here. We got it from our Moodle page. It just says class data. Oh, that's what it looks like, by the way, give you a view. So we got it from a previous student. It just says a previous class. Okay, so we got it from a previous class. So that it needs to go into our methods section to state that where we got it from. And then you'll need to make sure you're citing the workbook for the methods. We didn't just make up the methods ourselves. We need to cite the workbook as where we got our methods from. Okay. So that should only take a paragraph. Okay. Next, we have to give the results. So the results section is displaying the results, but we're not actually interpreting the results. We're just stating the patterns that we see or the trends in data that we see. But we're not actually making any formal conclusions or anything. So with the results, we open the results with an effective overall finding, not specific sets of data, but general trends that you saw. So basically, what I suggest is that you just start out with stating how many sets of data were we able to collect and what overall trends in data do you see. So let's take a look at the data. So first off, how many sets of data were we able to collect? Okay, so we have this set and this set. And this set, and this set. So we got basically 2 sets of data for the sodium bicarbonate and 2 sets for the distilled, right? So we ended up with collecting 2 sets of data for each type of water, I guess. And then you can just, based on that information, just generally state what you saw. Did you notice one particular wavelength of light seemed to have the most photosynthesis taking place? Did you see one particular wavelength of light see no photosynthesis taking place? So that's all you need to state is, we ended up collecting 2 sets of data for each type of water, and it appears that this wavelength of light showed the most photosynthesis, or maybe there wasn't any general trend in data, who knows. Okay, but just open it with maybe 1 or 2 sentences of the overall trends in data. Next, you're going to provide the visuals, okay? So in this case, we're going to actually include the graph. Now, remember we actually did a hand-drawn graph as one of our discussion forum posts. So if we, go back to our Moodle, in our discussion post, we actually did the bar graph. Okay, so we graphed the average of the distilled and the average of the sodium into one graph. Okay, so instead of having it in as a hand-drawn graph, we're going to put it in the old Excel spreadsheet and make it computerized. Now, if you don't know how to do that, here's a link, Graphing Using Excel. So this is a way that you can watch this, Kaltura video resource and see how you can make a graph, okay? So this way you can kind of, get an idea of how to graph that data that we, we hand drew that graph. Now you gotta make it into a nice, professional looking graph, okay? So once you make it into a nice professional looking graph, then you got to make sure that it's all together in one bar graph. So we're going to include the averages for both the bicarbonate and the distilled water. The x-axis, remember, will be the wavelengths of light. The y-axis will be the percent of spinach disc photosynthesizing after 30 minutes. And then you will have the bar graphs for each. Okay, so we'll have the bar graphs for the distilled water, bar graphs for this sodium bicarbonate. So there'll be 2 bar graphs for each wavelength of light. And then just make sure that we can distinguish the distilled water bars from the sodium bicarbonate bars. So you'll have a legend present. Okay. So all of that will be, in one single graph. Make sure there's a title to your graph. And then with your graph, you'll want to label it. So you can label it as graph number 1 or figure number 1, whatever you want, because you'll be referring to it later. Included with your graph, you'll need to write generally a paragraph describing the trends and data that you see in the graph. Okay, so what trends and data do you see? Do you notice, again, is there some wavelengths of light that are photosynthesizing more than others? Is there a type of water that is allowing photosynthesis to occur more than others, and so forth. So you'll need to write out the trends of data that you can see in that particular graph. And then additionally, in your text, you're going to need to incorporate the number of the graph included in the actual text. So reference the table or graph by number. So for example, in the text you can say, according to graph number 1, the green wavelength of light seems to exhibit the least amount of photosynthesis or the most amount of photosynthesis, blah, 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 blah. So you're going to reference the table within the text. Okay, so that's part of the rubric. So you need to successfully integrate verbal and visual representations. So reference the graph and make sure you number the graph. All right, and then finally we get into the discussion. So the discussion is where we're going to actually interpret the data. So in the results, we're just explaining the trends in data. We're just observing the trends, we're stating them. Now we're going to explain them. So we're going to actually interpret them. So the discussion, we're going to start out by opening with an effective statement of support of hypothesis. So what was the hypothesis and was it supported by the data? So basically you're going to start out with, what was your hypothesis for the wavelength experiment? And then do you formally accept or reject it based on the data? And then why or why not? And then same with the hypothesis for the distilled versus bicarbonate experiment. Do you formally accept or reject it based on the data? And then why or why not? Okay, so that's the first part of your discussion. Next, you're going to back up the statement with reference to the appropriate findings. So if you look in your packet, your workbook, at the end of the topic, it actually gives you some things to address. So it says here, I included some of them in the intro, and then here it says use your data to describe how to increase. So this stuff you can put in your discussion section. Okay, so these questions you can answer in your discussion section. So use your data to describe how an increase in the atmospheric CO2 concentration might affect photosynthesis. So when we're looking at the distilled water versus bicarbonate, you noticed a difference. So based on that, how do you think an increase in our own carbon dioxide concentration in our atmosphere might affect photosynthesis? Will it be good for plants? Will it be bad for plants? So you can use what we gathered from this experiment and apply it to what's happening with our greenhouse effect, our increase in carbon dioxide emissions. And then the second part with the wavelengths of light, green plants do not absorb green wavelengths of light. So describe how your data supports this statement. Or maybe it rejects it. I don't know. It depends on the data. So, you can use your data to either, support or reject this statement. So this again gets included in your discussion after you're accepting or rejecting your hypotheses. Next, as part of the discussion, we want to effectively link answers of research question to solutions of problems, blah, blah, blah. So in terms of this, we want to describe the relationship of transpiration to photosynthesis. So what is transpiration? So transpiration is basically where plants, it's kind of like they're sweating, right? So their water is leaving the stomata of their leaves, and then the water evaporates back into the atmosphere. So how is that related to photosynthesis? So describe that relationship. And then you'll also want to describe the relationship between photosynthesis and primary productivity. So you're extending the results to the real world. So what happens if, photosynthesis, if the rate of photosynthesis declines? How's that going to affect our primary productivity? Okay. So those are kind of, expanding the experiment to other applications. And then finally, at the end of the discussion, this is where we're going to kind of address any issues pertinent to the lab. So for this experiment, we're going to look at what were some sources of error in this experiment. So even though we didn't actually conduct this experiment, think about what were some sources of possible errors in this experiment. So we did see a picture of the experiment with the student data. Okay, so you can kind of see how the experiment was set up. So, what were some possible errors, do you think, of how this was set up? Look, here's the lamp, here's, the little color foils that they placed over it. Do you think there could have been some possible errors with the way this was set up? So think about some possible errors. Also, remember, we only had 2 groups worth of data, so could that have been a source of error? And then if you were to change the experiment, describe what you might do differently to give you more consistent or reproducible results. Okay, so if you looked at the averages, they were, or the data itself, the raw data was kind of a little bit skewed, wasn't completely, the same for both groups. So what could we have done to get more consistent data? So think of that when you're writing this section. So all of this together composes the discussion. And then finally, you'll have your conclusion. So your conclusion is basically telling the reader, what did you learn, what surprised you, and what will you remember for the next lab? So the first part, what did you learn overall from the experiment? So what did you learn about light and photosynthesis? And then think about what surprised you about it. And then finally, what will you remember for your next lab activity? So sometimes it's, since we didn't actually conduct the lab, think about like the whole scientific method part. Some of you, it might be pertaining to the actual graphing part. Some of you, it might be pertaining to how to write the lab report. Okay. And then the reference section. So where did you get your information from? So you need to cite the workbook, since we got our methods from that. You should probably cite the textbook, since you'll probably get a lot of information from that. And then to get a high, get the maximum points for the reference part of the rubric, you'll want to cite another source. So you need to have at least three plus references to get full points for that part. Make sure that all of your references are in APA format. So you might need to use citing your references in the library, or you can just use one of those websites on like how to cite APA to help you cite properly. Make sure you're including in-text citations throughout your report when you're using information obtained from a source. So they go at the end of the sentence in parentheses, and it's just author, year. Okay. And then presentation and grammar. So basically what I look for is it easy to follow? So you should be having section headings in order. So like I put in this PowerPoint, okay, each of these section headings should be included in your report. So you should have a methods section heading, and then your methods section. You should have a results section heading and your results section under it with the paragraphs and the figure. Discussion should be a section heading. Okay, conclusion should be a section heading. So everything should be easy to find. Everything should be in paragraph format, so it's not in like bullet points or anything like that. Make sure you indent your paragraphs and you're just following like the normal, like how to write a essay type thing with 12-point font. You don't have to use Times New Roman, but it should be like something similar, double spaced. Make sure you use spell check and grammar. Read it, proofread it, that kind of thing. Okay. And then once you are all ready to submit, don't forget to grade your paper, okay? So, let me go back and change the student role. When you go to submit your submission, okay, before you click add submission, go below and grade it, okay? Make sure that you get, check marks in all of the categories on the right hand side. So do you think your title is consistently and accurately describes the lab? Do you think your abstract consistently and effectively summarizes? Did you effectively define the research problem with elaboration? Did your hypothesis and reasoning present with elaboration and reasoning and consistent? Okay. Did your method section include the workbook as cited? Okay. Things like that. I always have students miss this one. Overall findings effective and separate from the detailed description of the results. Okay. A lot of students forget to do this one, multiple sources of errors and many future possible experiments. So grade yourself. I hate giving zeros because students simply forget a category here. So use this rubric to grade yourself before you actually submit. And by the way, at the end of this, it says image of student included. We did not actually do a lab ourselves, so you can ignore that part, okay? Everything else you should be doing. If you have any questions about this particular lab report, feel free to email me or visit me during my office hours. I'm here to help. Good luck, everyone.